Cancer remains one of the leading causes of death worldwide, underscoring the urgent need for therapies that are more targeted, less invasive, and capable of overcoming the limitations of surgery, chemotherapy, and radiotherapy. The rise of nanotechnology has created new opportunities in oncology, enabling treatments that operate with precision at the molecular and cellular scales. Among these emerging technologies, iron oxide-based magnetic nanoparticles have gained particular attention. They can be guided by external magnetic fields, enhance image contrast, transport therapeutic agents, and as demonstrated in both infectious disease and cancer research, serve as versatile biomedical tools. Most importantly for this project, MNPs can generate heat when exposed to alternating magnetic fields, a therapy known as magnetic hyperthermia. By raising tumor temperature in a controlled and localized way, magnetic hyperthermia selectively damages cancer cells while sparing surrounding healthy tissues. Because MNPs combine magnetic controllability, biocompatibility, and tunable physical properties, they represent a unique convergence of material science, physics, and biomedical engineering. This project examines their magnetic behavior, the mechanisms behind heat generation, engineering strategies that enhance therapeutic performance, and the current clinical progress and future directions of magnetic hyperthermia. Magnetic nanoparticles used in medicine are typically made of iron oxides such as magnetite or maghamite. At the nanoscale, these materials exhibit superparamagnetism, meaning they can rapidly align with the magnetic field but do not retain magnetization when the field is removed. This minimizes magnetic aggregation within the body. However, van der Waals forces can still cause clustering, and therefore nanoparticles require stabilizing surface coating, commonly known as PEG, dextran, or silica, to ensure dispersion and biocompatibility. MNPs also feature a large surface area to volume ratio, which allows scientists to attach functional molecules such as antibodies, peptides, or drugs. Once functionalized, these nanoparticles become versatile tools capable of tumor targeting, imaging enhancement, and controlled therapeutic heating. Through material engineering, their magnetic, chemical, and biological properties can be finely tuned, enabling the development of increasingly precise medical nanoplatforms. This last feature forms the basis of magnetic hyperthermia, where the nanoparticles act as microscopic heaters that can selectively raise the temperature of diseased tissue to damage or destroy it. The remarkable thing about MNPs is how they combine physics, chemistry, and biology into a single technology. From their magnetic response to their surface chemistry and biological compatibility, each property can be tuned through material engineering. As a result, magnetic nanoparticles have become a foundation for many emerging smart medical systems, ranging from rapid diagnostics to precision cancer therapy.
Once we understand what magnetic nanoparticles are, the next step is to explore how they actually generate heat to destroy cancer cells. This process is known as magnetic hyperthermia, and it relies on converting magnetic energy into thermal energy at the nanoscale. When magnetic nanoparticles are placed inside an alternating magnetic field, the rapid changes in the magnetic direction cause internal friction-like effects that release heat. Several physical mechanisms are responsible for this transformation, but the dominant ones depend on the magnetic state, size, and local environment of the nanoparticles. In ferromagnetic and ferrimagnetic particles, the main mechanism is called hysteresis loss. As the magnetic fields alternate, the magnetic domains inside each particle continually reorient, creating a hysteresis loop in the magnetization curve. The area of this loop represents the amount of energy converted into heat per cycle. In superparamagnetic nanoparticles, which are smaller and contain only a single magnetic domain, heating is mainly produced through near and Brownian relaxation. In near relaxation, the magnetic moment flips direction inside of the particle in response to the external field. In Brownian relaxation, the entire particle physically rotates within the surrounding field, producing frictional heat. However, an important point for real biomedical applications is that the Brownian relaxation is largely suppressed inside the human body. Once nanoparticles enter dense environments such as intracellular compartments, which are endosomes and lysosomes, extracellular matrices, gels, or solid tumors, and the fluid around them become too viscous or physically restrictive for the particles to rotate freely. As a result, Brownian rotation becomes strongly hindered, and near relaxation becomes a dominant heating mechanism during actual cancer treatment. This distinction is cruel because it means that nanoparticles must be engineered within their internal magnetic properties optimized for near relaxation if they are to remain effective ones delivered into tumors. The overall heating performance is quantified by the specific absorption rate, which measures how much heat a given mass of nanoparticles produces per time under an EMF. SAR depends on particle size, magnetic moment, crystal structure, field strength, and frequency. Because excessive magnetic fields can also heat surrounding tissues, treatment conditions must remain within the biological safety limits. The best known guidelines is the Brezovich limit, which states that the product of the field amplitude and the frequency should not exceed about 5 times 10 to the power of 10 A over ms. In 2022, you and colleagues provided one of the clearest experimental demonstrations of these principles. They synthesized pairs of nanoparticles within the same size and shape but different magnetic states, allowing them to isolate the effect of each heating mechanism. Their results confirm that relaxation-based heating in superparamagnetic nanoparticles is significantly more efficient under clinically safe magnetic field conditions than hysteresis-based heating in larger ferromagnetic particles. This kind of research bridges fundamental physics and biomedical engineering, helping scientists design nanoparticles that heat precisely and reliably inside living tissues. By understanding the mechanisms that dominate inside real biological environments, engineers can move from basic magnetic materials to medically viable cancer therapies.
The next step is to design magnetic nanoparticles so they heat effectively and safely inside the body now that we know the physics underlying magnetic hyperthermia. The objective is to produce as much heat as possible inside tumors while adhering to clinical safety regulations. Particle form and structure are important design factors since magnetic behavior is heavily influenced by geometry. Shapes like cubes, rods, and octahedra have more magnetic anisotropy, which frequently results in better magnetic heating performance, even if spherical nanoparticles are the simplest to create. The multi-core or nanoflower structure, in which many magnetic cores group together inside a single particle, is another effective design. Under therapeutically safe magnetic field intensities, their linked magnetic moments improve specific absorption rate and increase magnetization, enabling effective hyperthermia. Another effective engineering tactic is doping. Researchers can adjust the crystal lattice and alter magnetic anisotropy, coercivity, and saturation magnetization by partially replacing iron with metals like manganese, zinc, or cobalt. For instance, under clinically safe fields, cobalt-doped ferrites typically show stronger magnetic responses. Additionally, biological behavior and treatment efficacy are significantly influenced by surface chemistry. Coatings like PEG, dextran, or chitosan increase colloidal stability, decrease immune recognition, and prolong circulation duration because bare iron oxide nanoparticles have a tendency to agglomerate or oxidize in physiological fluids. More sophisticated shells can provide multimodal functionality. Gold shells allow nanoparticles to function as dual-mode agents for both magnetic and photothermal therapy, while silica coatings allow chemical modification for drug loading. Therefore, simultaneous hyperthermia and NIR light heating are made possible by gold-coated MNPs, boosting temperature rise at lower field strengths. Both surface charge adjustment and size are crucial. The kidneys quickly eliminate particles smaller than 10 nanometers, while particles larger than 100 nanometers frequently build up in the liver and spleen. According to studies, the ideal therapeutic sizes are between 10 to 50 nanometers, which balances heating efficiency and circulation duration. In vivo biodistribution is enhanced by neutral or slightly hydrophilic surfaces, which reduce protein absorption. Every engineering decision has an impact on the magnetic, chemical, and biological behavior of nanoparticles, whether it is changing their form, crystal composition, or surface chemistry. Their medicinal effectiveness and heating efficiency can be greatly altered by even little changes. Because of this, magnetic hyperthermia is a very interdisciplinary field whose advancement depends on combining chemistry, material science, and biomedical engineering to create particles that function dependently inside the body. In general, SAR, targeting capabilities, and biocompatibility are directly impacted by every technical adjustment, including shape, doping, surface coating, and size. Magnetic hyperthermia has progressed from laboratory experiments to real clinical use, showing how physics and medicine can work hand in hand. The most successful and widely studied materials are iron oxide nanoparticles, IONPs, which are already approved for certain medical imaging applications and have entered clinical trials for cancer therapy. In magnetic hyperthermia, these nanoparticles are delivered directly into the tumor, either through injection into the tumor mass or systemic administration, where a portion of the injected particles may still reach the tumor depending on vascular and biological factors. Once inside the tumor, the patient is placed in an alternating magnetic field, AMF. The field penetrates deep into tissue and interacts with the magnetic cores, producing localized heat that raises the tumor temperature to about 42 to 45 degrees Celsius. This range is carefully chosen because it damages cancer cells, which are more heat sensitive, while largely sparing normal tissue. A notable clinical example is the MagForce nanotherm therapy, which achieved regulatory approval in Europe for treating glioblastoma multiforme. In this procedure, magnetic nanoparticles are injected directly into the tumor and activated by an external magnetic field to generate heat internally. The treatment is typically combined with radiotherapy, and clinical results have shown improved tumor control and prolonged patient survival compared to standard therapy alone. One of the greatest strengths of magnetic hyperthermia is its compatibility with other treatment modalities. Mild heating improves blood flow and cell membrane permeability, allowing chemotherapeutic drugs to penetrate deeper into tumors. It also increases DNA damage susceptibility, which enhances the effects of radiation therapy. Moreover, the controlled heating process can trigger immunogenic cell death, releasing tumor antigens that help the body's immune system recognize and attack remaining cancer cells. For clinicians, being able to see where nanoparticles accumulate is just as important as heating them. Iron oxide nanoparticles are widely used as MRI T2 contrast agents, enabling visualization of particle distribution before treatment activation. More recently, magnetic particle imaging, MPI, has emerged as a powerful technique to visualize and quantify nanoparticle distribution in real time. This enables image-guided hyperthermia, where the magnetic field can be adjusted based on feedback to ensure uniform heating and prevent damage to healthy tissue. Finally, safety and biocompatibility remain essential for clinical success. Iron oxide nanoparticles degrade into metabolically usable iron ions inside the body, while coatings such as PEG or dextran help reduce immune clearance and oxidative stress. Altogether, magnetic hyperthermia represents a promising addition to the oncologist's toolkit. It complements existing cancer therapies, provides imaging feedback for precision control, and demonstrates how engineered materials can operate safely inside the human body.
Magnetic hyperthermia has developed from theory into a recognized experimental and therapeutic strategy throughout the last 20 years. Research demonstrates that magnetic nanoparticles can specifically destroy cancer cells, provide regulated heat in living tissues, and improve other treatments. Because of their great magnetization and biocompatibility, iron oxide nanoparticles Fe3O4 and gamma Fe3O4 are the most researched. They heat quickly under alternating magnetic fields, establishing a stable thermal equilibrium that serves as a self-limiting safety mechanism according to early research. Tumor targeting and biocompatibility are enhanced by surface changes such as protein passivation or coating like PEG and folic acid. These particles can locate tumors, improve MRI contrast, and provide therapeutic warmth according to in vivo investigations. Aminosilane coated nanoparticles have been used in clinical trials to safely heat brain tumors to 42 up to 49 degrees Celsius, which has led to the approval of treatments like MagForce Nanotherm for glioblastoma in Europe. Nanoparticle accumulation within tumor cells has been visibly validated by imaging techniques such as the TEM and the EDX. In order to balance efficient heating with safety, the field now operates within predetermined limitations, usually magnetic fields of 6 to 30 kA per meter and frequencies of 60 to 500 kHz. All of these developments demonstrate that magnetic hyperthermia is a clinically proven, repeatable, and adjustable cancer treatment.
With these milestones achieved, the field now faces new challenges and opportunities. Today, most research on magnetic hyperthermia is focused on refinement rather than reinvention. Scientists are no longer trying to prove that magnetic nanoparticles can heat tumors, they're working to make this heating more precise, predictable, and even personalized. One major area of interest is temperature control. Since iron oxide nanoparticles do not naturally self-regulate their temperature, current research focuses on engineered strategies to stabilize and control heating, such as feedback-guided field modulation or nanoparticle coatings that respond to local thermal conditions. Another key challenge under investigation is the protein corona, the layer of biomolecules that forms around nanoparticles in the bloodstream. Studies are looking for stealth surface coatings that either resist or manage this layer, so that particles can circulate longer, avoid immune clearance, and retain their magnetic response once inside the body. Researchers are also working toward international standardization. Different laboratories currently use different field strengths, frequencies, and calorimetric setups, which makes comparing data difficult. Establishing shared SAR measurement protocols and reference materials is now a major priority for ensuring reproducibility across clinical trials. Finally, the field is moving toward multimodal treatments, systems that integrate hyperthermia with other treatment and imaging modes into unified feedback-controlled nanoplatforms. Coupled with real-time tracking through MRI and emerging tools like magnetic particle imaging, these hybrid systems could eventually deliver heat, drugs, and image contrast in a coordinated way, bringing magnetic medicine closer to true precision oncology. Looking beyond current research, we imagine a future where magnetic nanoparticles evolve from passive heat generators into active, intelligent agents inside the body. One idea is to develop magnetic nanoparticles with built-in sensing and decision-making capability. By integrating temperature-sensitive or pH-responsive layers, they could autonomously adjust their magnetic response depending on the local tumor environment, heating more where the tissue is hypoxic or acidic and less in healthy regions. Another concept is magnetically guided self-assembly, nanoparticles that can reorganize themselves once they reach a tumor, forming dynamic networks to maximize local heating efficiency before disassembling for safe clearance. Controlled magnetic interactions and relaxation mechanisms have already been described experimentally, but the self-assembly behavior described here remains a future direction. We also see potential in energy-coupled therapy systems, where magnetic hyperthermia is paired with nanogenerators that harvest the temperature difference to power microsensors or drug release mechanisms. The underlying principle of thermally responsive nanomaterials exists, while the integrated system we describe here represents a possible future application. Beyond medicine, these same principles could inspire engineered smart materials, surfaces, or coatings that self-repair, change stiffness, or release molecules in response to magnetic stimuli. Magnetically responsive materials have been discussed in nanomaterials research, but extending them into adaptive multifunctional systems is part of the long-term vision. Throughout this project, we explored how magnetic nanoparticles, though only a few billionths of a meter wide, can have a massive impact on how we detect and treat disease. By combining the principles of physics, material science, and biology, they represent one of the most exciting frontiers in modern biomedical engineering. We began by understanding the fundamental properties of these particles. Their superparamagnetic behavior allows them to respond instantly to magnetic fields and just as quickly lose magnetization when the field is removed, which makes them both highly controllable and biocompatible. This property forms the foundation for their use in medicine, from imaging and targeted drug delivery to the generation of localized heat. We then looked at the mechanisms behind magnetic hyperthermia, where alternating magnetic fields are used to make these nanoparticles vibrate, flip, or rotate at the nanoscale, releasing controlled heat that can selectively destroy cancer cells. Studies that have been cited have shown that particles dominated by relaxation losses in the superparamagnetic state perform best within safe clinical limits, confirming the path forward for future designs. From there, we examined how careful engineering and optimization, such as controlling shape, size, composition, and surface coatings, directly affect performance. These design strategies determine not only how efficiently the particles heat, but also how well they circulate, target tumors, and remain stable in the body. Clinically speaking, magnetic hyperthermia is already proving its potential. The MagForce nanotherm therapy for brain cancer demonstrates how targeted magnetic heating can complement radiotherapy to improve treatment outcomes, all while minimizing harm to health and healthy tissue. It's an approach that blends precision, safety, and adaptability, qualities that define the future of personalized medicine. Ultimately, the story of magnetic hyperthermia is one about engineering heat at the nanoscale, turning magnetic energy into a medical tool that can both diagnose and heal. The future now lies in refining these materials, improving efficiency, and integrating imaging and control systems to make treatments even smarter and more reliable. By mastering the science of magnetic nanoparticles, we move closer to a world where medicine is not just reactive, but engineered for precision, allowing us to fight diseases like cancer with accuracy, intelligence, and hope.